Good afternoon, everyone. Good morning if you're on the West Coast. My name is Stephen Roach. I'm the director and principal consultant for ASR Consulting, Animal Health and Welfare Research Consulting Group that is working with the Dairy Farmers of Canada and researchers across the country to coordinate a series here we're calling the Virtual Research Showcase. And what we're here to do today is to dig into the details and the updates and the science and the stories that are coming out of that research on a particular project here that's really focused on a variety of angles around antimicrobial stewardship. Today is going to be the second event in a series of about 15 to 20 virtual showcases that things are still in development as we look into 2023, because there's been quite an interest in doing a number of these. We're looking to highlight dairy researchers and dairy research projects from across Canada, as I mentioned. So additional details are going to continue to come out and will be shared towards the end of the showcase for future meetings and future topics as well. And I would want to remind everyone here that we're hosting this event as more of a traditional Zoom meeting, which means you have the ability to turn your cameras on, your microphones on. For best audio quality, we do ask that you please keep yourself muted until you're called upon for a question or a comment if you are. We're going to take questions in the chat. So please send your questions to my teammate Carly or myself in the chat box below as they come up, and we're going to do our best to address those questions in the question and answer period that we have towards the end of the session today. Anything we don't have answers for in the time we have allotted, we're going to follow up after the fact with and get some responses and share those out after the fact. For those of you that would like to hear this event in French, you can access the French interpretation feature by clicking at the bottom of your screen on the interpretation globe. Select the French language option and mute original audio. That will give you access to our French interpreter who's going to be alongside us for each of the presentations. If at any point you need any assistance from any of the technical features of the meeting or questions, please also feel free to contact us in the chat, specifically to myself or to Carly. I now want to hand things over to a representative staff member of the Dairy Farmers of Canada, Amy Desolate, for a quick introduction and a thank you from DFC before we begin with our featured speakers. So Amy, you want to take it away?
Thank you, Steve, for this introduction. So my name is Amy Desolate, and I'm Assistant Director Research at Dairy Farmers of Canada. I'd like to thank you all for registering for today's webinar. As mentioned by Steve, this is the second edition of a new webinar series launched by DFC just a couple of weeks ago as part of our ongoing commitment to research, communication, and knowledge translation. This webinar series aims at showcasing the exciting work happening in dairy research across Canada. This initiative is possible thanks to the financial support of DFC, but also to our partners. And I also want to thank the research teams from today, but also all the ones from across the country for their collaboration in their initiative. And thank you as well to ASR Consulting for essentially organizing everything. So without further ado, I will turn the floor over to you again, Steve. Thank you.
Great, thanks, Amy. And again, thank you to Dairy Farmers of Canada for really supporting this and helping us get this type of activity, as well as a number of other ones we're excited to share as the year unfolds and we get into the new year with everyone as well. So we are going to kick into our official program in just a moment. Before we do that, I do want to just introduce our two primary investigators for a project that has been funded as part of the Dairy Farmers of Canada cluster initiative here, and that's Dr. Javier Sanchez and Luke Heider. Javier, would you like to just make some opening remarks here for everyone before we jump into some more focused presentations?
Yeah, sure. Thanks, Stephen. Hello, everyone. And my name, as Stephen mentioned, is Javier Sanchez. I'm epidemiologist at the Atlantic Veterinary College. And just a few words before the most important people on this day take over the seminar or the webinar. First of all, I guess it's a pleasure to be here and to show you or summarize some of the main research funding that we accomplished over the last 4 years. This project started in 2018. I guess that's when we initiated some of the conversation, and then proposed, was submitted, and then by 2019, we're on the field sampling these 150 dairy farms across the country. And this, by no means, I'm a co-investigator with Luke, but this is the result of a joint effort of all the veterinary school, Memorial University as well in Newfoundland. But I guess a key role in this project has been the Public Health Agency of Canada, led by Dave Leger and Daniela Rizzo, that help with a lot of the logistic of organizing and coordinating all the sampling, and be able now to summarize and present you some of these results. So we're at the end of the project. Is finishing in before next March. We're in the phase of organizing the final face-to-face meeting with all the industry, vets, and potentially will take place in Guelph in sometime between January and February. We're finalizing those dates, and as soon as we have those, we'll communicate that. So I guess that's my few remarks, and hopefully we can answer some of your questions. And I guess I'm gonna pass it to Luke and see if you wanna add something else, Luke, and before we keep going.
Sure, and I don't have very much to add, but my name is Luke Heider. I'm a dairy clinician here at the AVC, and also have a PhD in epidemiology. Again, just thanks to everyone who's been a part of this, with a number of collaborators across the country. It's been a real, I guess, pan-Canadian effort to try to get this surveillance system put forward, and so it's been a lot of hard work. So really appreciate everyone. Also appreciate the graduate students. You know, without them, a lot of this research wouldn't get done, because they are the drivers of getting this completed. So I'm happy for them to have a chance to share their research findings and what they've found. So anyway, without any much further ado, I'm going to hand it back to Stephen. Thanks again for everyone for coming and watching and participating.
Thanks, Luke. So we're going to jump right into our first presentation. I'm going to hand it to my team member, Julia Saracini, to do an introduction of our first of several speakers. Julia, do you want to take it away?
Sure. Hi, everyone. Again, thanks for coming. Our first speaker today is Landon Mortar. So Landon earned his undergraduate degree in biomedical science and nutrition at the University of Guelph before moving to Prince Edward Island for graduate school. His work centers on quantifying antimicrobial use on Canadian dairy farms and investigating how that interacts with animal health. So I'll hand it to Landon, and very excited to hear your presentation.
Thank you. Really appreciate it. It's nice to have the opportunity to share this stuff with you guys. So yeah, I'm Landon. I'm PI, and I'm going to be talking about changes we've seen. So I'm sure everyone here knows that antimicrobial use has been under increasing scrutiny in dairy industry and animal agriculture at large. That's coming from government and pan-governmental organizations down through consumers and industry groups. So if we're going to be paying more attention to antimicrobial use, it's nice to see how things have changed. So in 2007 and 2008, there was an effort to quantify antimicrobial use on Canadian dairy farms using a garbage can audit, which I'll get to what that is in a couple of slides. But that provided us with a baseline. And from that, we could build on that using stuff from the CaDNet project, expanding on some other work done with Quebec to do more of these garbage can audits across Canada and expand this network. And yeah, it's allowing us to see how things have changed. So the main goal here is to determine what antimicrobial usage looks like on Canadian dairy farms today, and then also to look back and see how things have changed since 2007 and 2008. So I've mentioned what a garbage can audit is a couple of times here, so I'll just go into some more detail. So I have some pictures here from a garbage can audit that was done with the project. You leave a garbage can on a farm and you ask everyone when they're empty out a medication of antimicrobials they've been using on the cows to just toss it into the bins here. And then someone comes back at regular intervals to count everything that's in there. And that's a pretty decent way of measuring antimicrobial use on a farm. So this was how antimicrobial use was quantified in 2007 and 2008 in Alberta, Ontario, Quebec, and Nova Scotia. And then Quebec did a follow-up garbage can audit in 2018 and has since moved to some other methods I'll get to a bit later for quantification. And then CaDNet built on with doing garbage can audit in British Columbia, Alberta, Ontario, and Nova Scotia in 2020. So we have follow-ups here from 4 of the provinces, everyone except for British Columbia, where we just have the more recent results, but I'll still be talking about how their usage fits into this as well. So it's fine and dandy to say that a garbage can audit measures antimicrobial use, but you can't just like talk about the number of bottles or the number of tubes, because that isn't going to give you a real good idea of how the antimicrobials are being used and the actual amount that's being used. So we're going to be talking more about dose-based metrics, which look at the therapeutic implications of the drug. So I'm specifically going to be talking about defined course doses per 100 animal years. With that, we're thinking about, if you look at the label on the drug, you can get an idea of how many treatments you can get out of that bottle or that tube. And from that, you can do some averaging between the different products to figure out the defined course doses. So we're just defining what the course is going to be. And then we're going to think about it in terms of 100 animals per year. So if you have a herd with 100 animals on it and you look at them for a year and you're measuring their antimicrobial use, is how many treatments of antimicrobials you're expecting to see on that farm. And then I built a computer program that takes that data from the garbage can audit, all the counting, and then converts it all the way over to the dose-based metrics for doing the analysis. And I'm going to talk about that at the end as well. So to start digging into the data, right here I have sorted all the farms. So the lowest use is on the left and the highest use is on the right. This is for all antimicrobials on the farm. Then on the y-axis, the height of the bars, that's measuring their use in DCD per 100 animal years, so courses per 100 animals per year. And then this big blue line is the median. So half of the farms are above it, half the farms are below it. You can see from that blue line that it's below 100 DCD per 100 animal years. So that's going to be less than 1 course of treatment per animal per year, which I think that's a pretty good sign that most animals are going to be going without treatment every year. If we're going to be looking at one drug in particular, it's going to be ceftiofur, because it's in a class of drug called 3rd generation cephalosporins, which are really good drugs. They're like, I'm sure anyone who's been using products like Xanel or Spectramast, like they have low withdrawal times, which is great for the dairy industry, but they're also really effective at treating a lot of conditions. And being important to dairy is one part, and then they're also considered to be very high importance to human medicine by Health Canada and some other organizations across the world. So it's something that we want to pay a lot of attention to. So again, I have sorted from least use to most use, from left to right, and then same again, DCD per 100 animal years on the y-axis. So you can see that most farms are using very little. There are 5 farms that use none, but most of the farms are using a very small amount of ceftiofur per year, which is pretty good to see. But if you go all the way to the right, you can see that there are some farms that are using substantially more. A lot of this comes down to blanket dry cow therapy. And that's one step. If you're looking at moving your farm farther to the left, it's a good place to start, is just looking at how you're using ceftiofur or whatever other drug you're concerned about in dry cow therapy, because that can stack up pretty easily. Now, that was what usage looks like in 2020. If we're going to be comparing that back to the 2007 and 8 studies, it's you get a pretty nice picture here. So across the 4 provinces where we have 2 points to look at, there was a substantial decrease in antimicrobial use, which is just great to see. That's going to be coming from some regulatory impacts, that's coming from industry priorities, it's going to be from consumer interests. There's a lot of factors going into that, but it's added up to a pretty substantial decrease. British Columbia, we don't have the 2 data points for, 2 time points. So we can't say for sure if it dropped there, but the other ones we can say that there's a pretty substantial drop, and it is statistically significant across them all. If we go to ceftiofur again, a drug that we're awfully concerned about, we're not seeing that trend. It looks like it has increased. A lot of this comes down to just Spectramast, because we didn't have an intramammary ceftiofur product in 2007 and 8. Spectramast came on the market at the end of 2008, so it was after the garbage can audit. So that right there just added a new category of ceftiofur to the market, but it is a drug we care an awful lot about. So Quebec has added some regulations onto it to try to reduce the usage of these very high important drugs. And that seems to have been quite effective in reducing usage. So moving forward, we're going to be moving away from garbage can audit because they're really cumbersome and slow, and towards using veterinary dispensing records, since all drugs or all antimicrobials for use in cattle now require a veterinary prescription. Just consolidates it, makes it a bit easier. And I've retrofitted my computer program to work on dispensing records as well. And this will allow for more continuous evaluation and just relieving some of the workload for quantifying antimicrobial use. And then we'd like to improve on some of the benchmarking that we can do for farms and provide farms and vets with some better data and interactive data so they can understand their usage a bit better. So in conclusion, it's great to see that we've seen a pretty significant decrease in antimicrobial use since 2008, but cephalosporins aren't following that trend. So that's something that's going to be worth attention in the future. Thank you.
That's great, Landon. Thank you so much. I also just want to highlight, Carly, our colleague, is going to add the French version of these slides in the chat. So anybody listening on the French channel, or if you prefer to view these slides in French, we also have that available. So you can find the link to that in the chat in just a moment. So our next speaker today is Tamaki. So Tamaki Uyama is a postdoc scholar in the Department of Population Medicine at the University of Guelph. Her research focuses on antimicrobial use and management practices in young calves on Canadian dairy farms. Go ahead, Tamaki.
Thank you very much, Julia, for your introduction. My name is Tamaki, and I'm very grateful to have this opportunity to explain a little bit about my research on antimicrobial use in pre-weaned dairy calves. So I would like to start off with why calves are important and why they matter. Studies show that most of the antimicrobials used on dairy farms is used for mammary health in cows, but some of them say that the calves are also commonly treated with antimicrobials. So the figure on the left shows that shows the result from the US-wide study, which showed that 76% of calves with diarrhea were treated with antimicrobials, even at a higher proportion in pneumonia cases. And these are the two major diseases seen in calves. Antimicrobial use is one risk factor to select resistant bacteria and elevate the level of antimicrobial resistance. I would also like to highlight that what kind of antimicrobials being used is really important to consider. So based on Health Canada's standards, they have categorized from very high importance, so category 1 to category 4, lower importance of veterinary antimicrobials based on human infections. And that leads to my objective to describe and quantify antimicrobials used in heifer calves on Canadian dairy farms. So for the materials and methods, we had 147 dairy farms enrolled across 5 provinces during the farm visits last year. For my research specifically, I looked at calf treatment records. So what I did was to basically look at the newborn heifers during a 1-year time period and follow them up to 60 days of age. I looked only at the heifers, and no beef calves were included. The data that was used for future analysis depended on the information that was available on the farm, and I will explain this further in the next slide. So as a result, out of 147 farms, I was able to retrieve the complete set of calf records that I needed from half of those farms. And there are a couple of reasons behind this. One is, as you can see on the pictures on the right side, some of the handwritten recording systems was quite difficult to keep track of. Some were ripped off. They had different formats, and some of them were non-permanent, so it could be weird during the period of time. But fortunately, I was able to have data from 74 farms, which resulted in 7,817 newborn heifers being followed. Among those, about 30% of them were treated at least once with antimicrobials, which was quite close to a study that was done in US. And among the reasons that was the antimicrobials that we used was mostly respiratory disease followed by diarrhea. And these were also the two major diseases found in calves. When looking at the different categories of antimicrobials that have been used, more than half of them were category 2 antimicrobials, so that was of high importance, followed by medium importance to human health. And you can see here that the very highest importance to human health standards were only used in 5% of the data that I was able to retrieve from the farms. So to summarize, category 1 antimicrobials were used less commonly compared to lower importance for human health in calves. And for future direction, I would like to raise awareness that having good records of antimicrobial treatments as well as other data in calves, too, would be really essential to keep track of how the dairy farming has improved over the years. And it will be nice to have those data. If any kind of electronic or management systems is been already adopted on the dairy farm, I would really like to have that being used for calves as well, in terms of having easier accessibility to the data in calves. That is the end of my presentation. I would like to thank all the dairy producers and people who led the project and helped me retrieve this data. Thank you very much.
That was great, Tamaki. Thank you so much. Again, just a reminder that we'll be taking questions for all the presentations at the end. So if you think of questions as they come up during each presentation, feel free to message the ASR colleagues with your question, and we will get to all of those towards the end of the presentations. Our next presenter is Dr. Mariana Fonesca. Mariana is originally from Brazil, where she completed her DVM and MSc degree in veterinary science. Currently, she is a PhD candidate in the Health Management Department at the Atlantic Veterinary College in PEI. She collaborates with recently implemented surveillance systems for AMU and AMR in Canadian dairy farms. And Mariana, we're excited to hear your presentation.
Okay, thank you, Julia, for the introduction, and thank you for the opportunity to talk today. And I will be talking a little bit of the antimicrobial use and association with resistance to ciprofloxacin and tetracycline in Campylobacter. So for a short background, why is important to study about Campylobacter? In Canada, there is a high incidence of human infections that are caused by Campylobacter. And although poultry is the leading source, data from the United States show that more outbreaks derive from their source compared to poultry source. And moving to the resistance, a study published in 2017 that collected data in the province of Ontario in Canada showed that 71.4% of the Campylobacter isolates from livestock, they were resistant to at least one of the antimicrobials tested. So the objective of the presentation is to determine the proportion of antimicrobial resistance in the Campylobacter isolates, and also investigate the association between the antimicrobial use and antimicrobial resistance in these Campylobacter isolates. So our data were collected through CaDNet surveillance. The surveillance was implemented in 2019 and was developed by this expert panel from different veterinary colleges and public health agencies of Canada. And the surveillance enrolled 140 farms across Canada. And all the samples across these 5 provinces in Canada, and all the samples, they were sent to the Atlantic Veterinary College laboratory to be processed. So during the fall 2019, we sampled 140 farms, which gave us a total of 506 fecal samples. And we had 4 different sample sources: pre-weaned calves, heifers, lactating cows, and manure storage. And we collected 5 fresh pads on the floor from each of these 8 different aids to have this, what we call composite manure samples, and then the samples were analyzed in the laboratory. So in the left picture, you can see a selective media that we use to isolate the Campylobacter. And on the right picture, we can see a picture of the machine that we use to do the what we call antimicrobial susceptibility testing. So this plate you are seeing the machine, we have different antimicrobials there in different concentrations. And then the machine spread our Campylobacter sample on this plate. And then we can see which was the lowest concentration of a given antimicrobial that was able to inhibit the Campylobacter growth. And then based on this concentration, we can classify this Campylobacter if it was resistant or susceptible to a given antimicrobial. And then using these results, we build a model to be able to investigate the association of antimicrobial use and antimicrobial resistance. You can, next slide. So the antimicrobial use, Landon already explained what is a garbage can audit, how we collect this information. And then we collected the information from the garbage can audit, and we converted this to a metric that is the DC, we call DCD, defined course dose. That is a dose for a standardized completed treatment course on a standard animal, multiplied by 100 to get this rate of 100 animals per year. Okay. So this figure is the results of that will show you the variability of the antimicrobial use among all of farms enrolled. So on the x-axis, we have the farms. So each green bar, it's one farm. And on a y-axis, we have the total DCDs per 100 animals a year. And I just add these two different lines so you can have an idea. The green line is the 25th percentile, and the red line is the 75th percentile. So if you click, yeah. So we can see here that all the farms here, the bars that does not touch the green line, it means that is the 25% of farms that was using less than 50 DCDs. And all the farms that trespass this red line, it means there was the 25% of farms that were using more than 147.6 DCDs. So higher users and low users, just to give you an idea on how these farms were using the antimicrobials. Moving to resistance results. So 71.6% of our herds, they had at least one resistance Campylobacter, and 53.8% of the Campylobacter isolates, they were resistant to at least one antimicrobial in the panel. And we tested these isolates to 8 different antimicrobials. So this figure here shows the resistance pattern by the sample source. Now we can see the different colors, the different source. So the dark blue is calf, light blue is cow, dark green heifer, and light green is manure storage. We observe a high proportion of resistance to tetracycline, followed by ciprofloxacin and allylic acid, and just a small proportion of resistance to azithromycin. And the other 4 drugs we tested, there was no resistance. And if we talk about antimicrobial classes, this represents resistance to 3 different classes. There would be quinolone, that is represented by ciprofloxacin and allylic acid, a tetracycline, and macrolide, that is the azithromycin. And 15.6 of these isolates, they were resistant to both quinolone and tetracycline. Now, moving to the results of the model. So like I said, we built a model to investigate the association of antimicrobial use and resistance to ciprofloxacin and tetracycline. I included here sample source just to show there was no difference of resistance among the different sources of samples we collected. However, for antimicrobial use, we found a significant association, but this association depends on which antimicrobial. So I will show a figure to be easier to understand. So this figure, you can see predict probabilities for resistance. On the y-axis, we have the predict probability of resistance to a given antimicrobial. That is in this case, we have the dashed line here that is for tetracycline, and the solid line is ciprofloxacin. So we can see that when we increase the antimicrobial use, that is our total DCDs, we can see that we increase the probability to have resistance to tetracycline. However, for ciprofloxacin, we got the opposite direction. And dive deep into the data to understand why was different for ciprofloxacin. So ciprofloxacin is a fluoroquinolone, And only 13 herds out of the 140 herds that we were investigating, they were using a drug that belongs to a fluoroquinolone class. And I just gave two examples here of the commonly fluoroquinolones used on dairy herds. So even a farm that is a high user, if it was not using a fluoroquinolone, this can explain why they have just low resistance to ciprofloxacin. As a conclusion, the overall resistance to tetracycline and ciprofloxacin was higher compared to the other antimicrobials we tested. And the total antimicrobial use was associated with an increased resistance for tetracycline, but not to ciprofloxacin. Like I said, this could be in consequence of the low use of fluoroquinolones in our enrolled herds. And for future steps, this is ongoing surveillance, like I said, was implemented in 2019. And with time, we will be able to analyze trends of resistance over time and also benchmark farms based on the antimicrobial use. So thank you, and I'll be glad to get the questions by the end of our presentations.
Thank you so much. Our next presenter today is going to be Ellen de Jong. Ellen is a fourth-year PhD student with Dr. Herman Barkema at the University of Calgary's Faculty of Veterinary Medicine. Her research focuses on the use of selective treatment protocols for clinical mastitis treatment decisions. So Ellen, feel free to start.
Yeah, thank you.
Ellen, I'm sorry, your audio, I cannot hear you. I'm not sure if that's the experience for anyone else or if you're able to change your audio here for us.
Is this working better?
Yeah, thank you.
Okay. Thanks for pointing that out. Yeah, no, I just wanted to highlight that the work that I'm presenting is also part of Katie McCubbin's PhD, and we're both supervised by Dr. Herman Barkema. So, as others have already touched upon parts of the CaDNet surveillance system, such as measuring antibiotic use, what Landon has presented, measuring antibiotic resistance, what Mariana just presented. And then there's a third part, the additional research projects, such as Tamaki's project, our project, which is focusing on, you can go to the next slide. Yeah, so we have focused on antibiotics used at drying off and antibiotics used for the treatment of clinical mastitis. And why have we focused on this specifically? Others have already mentioned it, but it is well, most antibiotics used on dairy farms are being used for treatments in the udder for clinical mastitis or during the dry period. So as illustrated here in red and also on this slide in a more recent study in Quebec, the same that Landon had referenced. So, as others have also mentioned, the antibiotics that we use to treat cows are very important to human medicine. And I've just listed a couple here that are being used for clinical mastitis and dry cow therapy. And I'm aware that not all of them are currently available to dairy farmers, but I also want to just pull your attention to the lines that says highly important and critically important, which really highlight the importance to human health, and why there's a big push to decrease the use of these antibiotics specifically. So our main question was how to reduce mastitis-related antimicrobial use. And there has been some trends in the last couple of decades that have made it possible to adopt a more selective treatment approach, such as a lower infection pressure of contagious bacteria, and I'm speaking specifically about other pathogens, and the availability of rapid diagnostic tests and also on-farm testing options. So what that means is that for dry cow therapy, we can now adopt a selective approach where we identify cows that would benefit from antibiotics at dry off. And this is not a new story. This is something that you've probably heard over and over and over again over the last couple of years, but it's still very important that we focus on selecting cows. And I will, the next couple of slides will explain that a little bit further. For clinical mastitis treatment, instead of identifying cows, we want to identify cases that benefit from treatments. So here we're using rapid diagnostic tests to select those cases that would benefit from antibiotic treatment. So we surveyed the farms participating in this CaDNet program. And what we found is that on average, 35% of producers have adopted a selective dry cow therapy approach. And this was back in 2019 when we did the survey. So hopefully those numbers have even increased more. And as a reference, in 2015, only 11% of surveyed producers practice selective dry gut therapy. If you look at clinical mastitis treatments, 64% of producers across Canada have been adopting a selective treatment approach. I do have to give the remark that this is a treatment approach. We did not ask the producers what their specific definition of selective treatment is, and I will touch upon that a little bit later as well. So moving back to those selective dry gut therapy protocols, we found that 84% of producers base their decision on somatic cell count history with a threshold of 200,000 cells per milliliter, and they take typically the entire lactation into account. They also use clinical mastitis case history, mentioned by 76% of producers. Bacterial culture and milk production are being used, but not widely. And if we focus our attention, sorry, go back to the previous slides. Yeah, if we focus our attention to the graph on the right, we see that there's quite a spread in percentage of herd coverage as drying off, and that this can be influenced by different factors. But most importantly, of course, the protocol use that they use to select those cows for antibiotic treatment at dry off. THCO is very important if we talk about selective drying off practices. It is recommended that every single cow receives a teat sealant, or if there is a selection being made, at least those that do not receive an antibiotic treatment. So in our survey, 32% of producers did not use any teat sealants at all. And of the ones who used teat sealants, only 56% of producers applied them to all the cows. So 12% did that selectively and used somatic cell count history, clinical mastitis case history. And again, on the graph on the bottom right, you see that herd coverage at drying off of teat sealants varies widely. And this was actually quite an interesting finding because we did not expect that. We expected that producers either use them in all their cows or not use them at all, and not use them as selectively as we found in our sample. So onto clinical mastitis treatments. So we asked the producers who said that they use a form of selection method in order to select cows for antibiotic treatment. We presented them a list of these 10 factors and asked them how important they were. So what we found is that there were 3 different types of producers present in our sample of those 150 farmers in the CaDNet sample. We had producers that only based their decision on the severity of the case. Then we had producers that, besides severity, also based their decision on the culture results, somatic cell count, mastitis history. And then we had producers who used a very broader range of cow factors. And this really illustrates the variety in protocols that exist and the different things that play a role in that decision-making process. And I also just want to highlight again that what we define as selective clinical mastitis treatments is really using a form of rapid diagnostic testing to identify those cases that do or do not need treatment. And so not always the definition that's being used by producers is the one that we use in the literature. And that's something that's good to remember because language is very important. So stepping back, so we have selective dry cow therapy and we have selective treatment of clinical mastitis. Which farms should be implementing this? It's farms that have good record keeping, so keep track of all clinical mastitis cases, have frequent somatic cell count reports, have on-farm culturing available, or have a vet practice that offers very rapid diagnostic testing, and farms that are willing to invest in equipment and train staff. And then if we take another step back, where does this research fit into? So we know that these protocols, these selective protocols, have been or are effective and will not lead to any negative consequences. Many studies have demonstrated that over the years. Now we know what the Canadian dairy farmers are doing. We can use this information and future studies on dairies or motivators to hopefully increase the uptake of these selective treatment methods and thereby reduce antibiotic use, and especially those critically important antimicrobials. So I have a take-home message. I'm just going to go through this quickly. But selective practices are very feasible and safe to enact on Canadian dairy farms, and especially on farms with a low percentage of other infections, and bulk milk, somatic cell count, and very good record keeping. So again, part of the research group of Herman Barkema, and I'll take any questions at the end.
Thank you. So our last presentation today is from Dr. Nikki Miller. Nikki's a PhD student in veterinary sciences and epidemiology at the University of Montreal. She also held a degree in veterinary medicine since 2019. So I'll pass it off to Nikki for our last presentation, and then we can go on to audience questions.
So hello, everybody. Thanks, Julia, for the introduction. So today we'll talk about some of the results that on the study that is actually ongoing at the veterinary faculty at the University of Montreal. It is about a new regulation. So what is this new regulation actually? We can change slides. Yeah, thanks. So in the province of Quebec, since February 2019, we have a new regulation that restricts the use of category 1 antimicrobials in animal production. So we need to prove that antimicrobials of lesser importance are not effective before using a category 1 antimicrobial. So we can use a nonspirogram, for example. And it also prohibits their use for preventive purposes. So after the implementation of the regulation, the first thing we wanted to do to know is if there's a change in the use of category 1 antimicrobials. So we did a study about that. We published the result in last February. I didn't put the link here, but if you're interested, I can send you the link. It is called Evidence of a Drastic Reduction in Category 1 antimicrobials in the province of Quebec. So I'll show you some of the results. We don't have time, a lot of time, but I will talk about some of a few of our results. So the results are based on the cells of antimicrobials. We compiled the cells with a veterinary billing software called VetExpert. Actually, Ilan Lardet in a study showed that VetExpert is as good as a Garbage Can Audit to look at antimicrobial use. So here you have a graph of category 1 antimicrobial use since June 2016 to April 2020. The y-axis represents the number of DCDs by months in the province. So for every farm, dairy farms, the 3,500 farms that were in our study. So here you can see that before the implementation of the regulation, so before the major drop that you see here, the use of category 1 antimicrobial was between 15 and 20,000. And after the implementation of the regulation, we were at less than 5,000 treatments sold each month. So you can see with this graph, which is a descriptive graph, but there is a major drop in the use of category 1 antimicrobial. We did so some models, and the changes are obviously significant. So we can go to the other slide. Thanks. Here you have the same, almost the same graph. So, but we show the total antimicrobial use, the blue or dark blue or black line. In the darker green, we have the category 2 antimicrobial use, and in the light green, we have the category 3 antimicrobials use. Here, I just wanted to show graphically that we don't see an obvious change in the use of other category antimicrobials, nor in the total antimicrobial use. Actually, I didn't put the model here and the result of our model here, but we don't see a significant change in the use of other categories. Oh yeah, there's the French slides, sorry. So here, I did, the results are, here we show, sorry. Here, I just wanted to show that it's the maiden use of category 1 antimicrobials went to 0 after the implementation of the regulation. So here in the x-axis, we have the number of DCDs, or the number of complete treatments sold in a herd in a year. And each curve represents a certain period. So you have the post-ovulation period, the pre-ovulation period, and the adaptation period. I don't want to go in detail here, but you can read the article if you want to go to see how we separated our periods and why we considered an adaptation period. But what is interesting here is that it's a black line. It's very small here, but it represents the mesan use. So in the pre-ovulation period, if we click on it, we see that the mesan use of category 1 antimicrobial was 10 treatment by herd by year. After the implementation of the regulation, we were at 0, like I said earlier. So it means that 50% of the dairy herd, the 3,050, here we have 3,300 herds, didn't buy any category 1 antimicrobials. It's more than 70% of the dairy farms in the province of Quebec. We have almost 5,000 farms. So that was the main result of this study. After that, we did another study. If we go to the other slide, where we look at the barriers and facilitators of the implementation of the regulation. So actually, we did some semi-structured individual interviews with dairy farmers and vets in the province of Quebec to know if what we did 1 year after the implementation of the regulation. So we wanted to know the barriers and facilitators. So here you have the result, the teams, you can click. The teams that emerged from our interviews. So I don't have time to go obviously through all of them. So I will only talk about the major ones. So we can go to the next slide. So the first major barriers that emerged by most of the vet and most of the producers was the heterogeneity of the understanding of the relation. So actually, there's some part of the relation that were not clear for producers and vet. For example, there's a part of the relation, it wasn't mentioned if there's no other antimicrobials available, if there's no category 2 or 3 antimicrobials available, can we use a category 1 antimicrobials without doing a diagnostic test? So this goes with the second barriers, the difficulty to access to alternative antimicrobials. So actually, the relation was obviously put in place at the beginning of the pandemic, and after the implementation of the regulation, many antimicrobials were not available from suppliers of veterinary medicine. Two other major barriers that emerged was the delays of diagnostic tests. Actually, in Quebec, the samples had to go to laboratories. So the samples had to go from the farm to the clinic to the laboratories. So it can take several days before having an answer. And then another major barrier was the fear for animal health and economical consequences. So it was because we don't know actually what are the economical consequences and the consequences for animal health of reducing antimicrobials. And many producers were reluctant to change their practice regarding antimicrobial use. And I just wanted to mention a minor barriers that was mentioned by a few producers, but is interesting. It was mentioned by producers who live near Ontario and New Brunswick. They didn't want to apply the relation actually and change their practice because it felt that it was unfair regarding their neighbors who don't have the same regulation, but their milks go in the same tank milk. So thank you. There's obviously more things to say, but it will be for another time. Thanks.
Thank you. Thank you to all of our presenters today. I also just want to highlight the funding partners for this project. So this is a collective slide of all the funding partners who helped us support these student projects today. And so I'll hand things over to Steve to do a few questions before we wrap up here.
Great, thanks, Julia, and thanks everyone for listening and for all of our presenters sharing some of the results of their latest work, and really exciting to see how some of these things dovetail and how each project is sort of helping to build out our understanding of some of these items in the context of antimicrobial stewardship, resistance, use, and sort of this broader conversation around what do we do and where do we go, and frankly, what's happening. I'm going to pull together some questions or a handful of different questions that I've been receiving into one bigger one. And I'm going to open it up to all of our speakers here for perhaps some comments here, just given our time frame. We certainly see from your work that use is going to lead to an increase in resistance at some level, right? And certainly there's many different avenues we can take in terms of trying to meaningfully reduce antimicrobial use on farms. Some recent work that I've been involved in, and a couple of folks have mentioned it here, is a fair number of people have been skeptical about how much more can the dairy industry meaningfully reduce without compromising dairy cattle health, for example. So I wonder, I want to throw it out to our presenters here. What's the take-home message about our ability today, based on where we stand, based on our understanding of antimicrobial use, to continue to reduce use without compromising health and welfare? Any comments for our listeners out there in terms of our low-hanging fruit opportunities, where we really ought to continue to go, balancing use, balancing these as important tools to maintain health and welfare, but also understanding the important implications of, in particular, category 1s and 2s, but all of our antimicrobials that have a linkage to our human health counterparts. So I'll open that up to our speakers. Any immediate thoughts there?
I can comment on that, Steve. I think it's very important that, and I think all the presenters have shown that there is such a variety between farms, and I think that is where we can reduce. Some farms are really still treating the minimum and really cannot reduce any further, but there are farms that are really big users, and they can adopt selective treatment practices, be more mindful with the antibiotic use, and I think reduce the total amount of drugs being used in the industry in that way. I think, yeah, so the key is, I think, in the spread and the variety of all the farms.
Yeah, there's certainly farms that can still reduce use. There are fewer of them than there were back in 2007 and 8, which is great to see. But there's two parts to stewardship. There's reducing the amount of use and then just being prudent about which drugs you choose to use. So there's two steps to make, and every farm can just at least evaluate where they are and what steps they can take.
I also want to comment that it's a great question to consider about the health, not compromising the health of the animals by reducing the antimicrobial use, and some can be related to farm management practices. In calves, some studies focused on colostrum management, and the thing that I did in my research was to feed like transition milk to calves. Also had an reduced antimicrobial use. And the farms that adopted that practice. So farm management practice can be something important as well.
That's great. Thanks, Ellen, Landon, and Tamaki. Tamaki, I want to just kind of follow up on something that's come through in the chat here. We often hear when we talk about specific areas of dairy where we could perhaps further reduce use is in the context of calves. And so just following up with some of your research, where do you see that the biggest opportunity in the context of calves to reduce use?
Yes, I think colostrum is definitely the biggest thing that we could consider to kind of, decide the future of that calf's health at a really early stage of life. So optimal colostrum management can be something that could be revisited again. I think it is most of the people here will definitely know the optimal practice, but I also assume that there's a wide variety among farms as well. So yes, reconsidering and revisiting your management practices could be certainly a key factor to consider.
Thanks, Tamaki. One other question that's come up a number of times, and several of you have talked about dry cow therapy and looked at it in different ways, talked about it in different ways. Is selective dry cow therapy our lowest hanging fruit opportunity for reductions, meaningful reductions in antimicrobial use as an industry? And to follow up on that, just to be a bit tongue in cheek, is selective dry cow therapy achievable for everyone?
Yeah, I can comment on that. Sorry, yeah, I think it's really, I think that lower adoption rate, we saw 35% across our producers that are adopting a selective dry cow therapy protocol. I think that really illustrates that is where we can make a lot of improvements. And I think that also resonates with what Landon showed us with Spectrumos being responsible for such a high use on dairy farms. And not every farmer or not every producer will be able to adopt the selective dry cow therapy approach. That's really something you need to discuss with your veterinarian. It's really important that you have a low bulk somatic cell count, you've got good record keeping, you've got your mastitis under control, and those are all factors that play a role in deciding whether or not it's something that fits your farm. But I think that it fits a lot more farmers than we sometimes believe. And I also just want to highlight that there is this great tool with LactaNet that's now available. So everyone who receives the SSC reports, I think they are all, you have access to an additional feature that allows LactaNet to inform you if you are fit to adopt a selective dry cow therapy approach and which cows you should be selecting. I don't know the full details. Katie is really the expert on that part, but that's definitely a tool that I think is very useful for producers and can increase uptake.
Wonderful. Thanks, Ellen. We have more questions, but I think we're at our time. So thank you to all of our presenters here. And for everyone that's listening, please just hang on for one more minute. Julia's just going to share an update as to our next webinar and what we'll be covering on our next topic before we go.
Thank you so much, everyone, for participating today. If you're interested in attending our next webinar, we'll be highlighting research from Dr. Nancy McLean and Dr. Linda Jewell. Carly's going to share some information in the chat about how you can register for the upcoming webinar, also some information about how to access research from the current presenters. So if you'd like to attend our next session, we'd love to have you. That's going to be on October 27th at 1 p.m. Eastern. And yeah, we'd love to see all of you there. So please make sure you're registered. And if you have any questions, please feel free to reach out to the ASR team or the presenters today. We'll also be sending out a follow-up promotional email with the recording from today's video, as well as a link to register for the next session. So thank you all so much. And it was great to have all the presenters today. And we appreciate everyone who's able to attend.
Thanks, everyone. Have a great day.
Thank you.